अस्सलाम वालेकुम। क्या हाल चाल है आप सब लोगों के? खैरियत से हैं? टुडे आर गोइंग टू डिस्कस अबाउट द ट्रांसपोर्ट across the cell membrane. As you already know about the transport across the cell membrane is a very important and vital topic in discussing about the cell system, and to maintain the integrity of the cell should have transport of materials, ions and other things that are very much important to maintain the integrity and self existence of the cell. अगर आप वैसे देखें, transport is actually traveling. If you don't travel, you cannot exchange things, you cannot exchange or buy things from the different parts of the world. So the transport have a very vital role in our life, as well as it has also very important and vital role in the integration and existence of the cell. So transport substances have very much mandatory, and sorry for inconvenience about the messages. Transport of substances across the cell membrane very much necessary to maintain the normal functioning of the cell. Normal functioning of the cell, it is purely linked with transport across the membrane. Your transport across the cell membrane, it is very much important. As I already told you, the self existence and its normal functioning is directly linked to the transport of cell membrane, transport across the cell membrane. So transport across the cell membrane, transport across cell membrane. As we already know that lipid soluble substances, water and urea, lipid soluble substances, number 1, number 2, water, number 3, urea. These can pass easily through the lipid bilayer of the cell membrane. They can pass very easily. We already know about it. Lipid bilayer, lipid bilayer of the cell membrane obviously is nearly impermeable to lipid soluble substances, which are the lipid soluble substances such as ions, charged or polar molecules. This is number 2, charged, yeah, or polar molecules such as glucose. They can easily pass. Actually, lipid bilayer is impermeable to lipid soluble substances. Sorry for inconvenience. These are substances that are impermeable to lipid bilayer. They actually need a specialized protein or a special substance. I should change the color here. They need special substance that is called protein channel or protein, which may be number channel protein or number 2 a carrier protein, whichever the case may be. These can help the charged polar molecules or lipid insoluble substances to pass through by this. The previously I was talking about the lipid bilayer. ions charge, these are the impermeable, insoluble, sorry, lipid insoluble substances. These can only pass by a specialized type of proteins, channel, transport, a van, a lorry, that is called proteins. Proteins are maybe of two types, channel proteins or a carrier proteins. They can pass through these proteins, they can ride through this protein and enter into the cells as according to the need of the cell. Besides these proteins, there is a specialized pumping system that may be called an active pump mechanisms. These active pump mechanisms can also help lipid insoluble substances to get enter into the cells to perform different functions or different to produce different proteins to form different hormones as compared to the other sites where these hormones or proteins are needed. Large macromolecules are transported through the vesicles. Number four are the vesicles. Vesicles, when used when the size is too large for the cell, so there is a vesicle formed just like this, and it will engulf this type of large molecules, and it will eventually, sorry about the color, it will eventually become inside the cell. So this is the almost a just an overview, just a bird's eye view of the transport and how why transport is necessary and how the cell can transport the materials. Lipid soluble substances, water, urea easily pass. Non-lipid lipid insoluble substances, lipid bilayers, cell membrane, ion charge, which are not soluble that can pass through channel proteins, carrier proteins, active proteins, and vesicles for two large substances. Now we are came here know about the types of transport, that which medium or which types of transport can be lead to a movement of these substances. There are many types, such as we start with the most simple. Number one is passive, with no energy needed. Number one is active, where there is an energy needed, and vesicular transport. As I try to give you concept about the vesicular transport, that is done by endocytosis, exocytosis, etc. Vesicular transport. These are the three basic and main types of the transport that came across the cell. Now we start with the basic and the primary, that is passive transport. It contains number one diffusion, number two osmosis, as I already know told you that Passive transport is there is no requirement or no need of energy required for that. We'll start with the diffusion. Diffusion actually is a movement of molecules that move from higher concentration towards the region of a lower concentration without an expenditure of energy. From high concentration to lower concentration, no energy needed. It's done spontaneously. It happening to that extent when the concentration gradient reached or gain an equilibrium. Uh, what about the equilibrium? If we have much more gas molecules in that region here and less molecules here, the molecules movement will be towards the region of lower concentration. When the molecules become equal, there will be no movement. So when the equilibrium achieved on either side, there will be no net movement of diffusion of a molecules from higher concentration towards lower concentration. We have normal, very basic examples that we already knew about it. That is, one is if we have a room or box, or we can call it room because I cannot draw a huge room in my mobile. So if we spray or sprinkle here just a perfume, it will spread all along the room, and you will feel the fragrance if you are sitting here and talking on the phone, but you will feel this fragrance. And another example is if you take a beaker, sorry about my hand free drops suddenly. It contains water. Water should be like this, and having much, I have to fill it up. Huh? Having this beaker of water. Sorry about my drawing. And we draw a potassium permanganate, pink. If we draw this in a hair and leave the water due to water molecules, this higher concentration will spread all along the water and convert it into a pink or a red color solution until an equilibrium is reached. Now the eight okay. What are the factors that affect the rate of diffusion? If we determine or want to know the factors, whether the diffusion is very much fast or diffusion is slow or diffusion is at once, it depends upon the factors. As I already mentioned about the perfume example, it is sprinkled in one corner of a room and then it eventually spread in a seconds or minutes to all over the room. But if we drop uh, a colored solution in a water, it will spread on the whole of the water, taking hours to a day. So these are the factors that eventually affect determine the effect of rate of simple diffusion. What are the factors affecting the rate of diffusion? So what would be the factors that can affect the rate of diffusion? Number one, concentration gradient. What it means, concentration gradients. It means the greater the difference between the higher concentration and the lower concentration. If we have molecules here much more and here a little bit and here again and another example. So the rate of diffusion in this pattern, number one pattern, this would be greater, and in this pattern would be slower because the concentration difference between from one region to another region, in the first case it would be higher and there would be more diffusion and there would be higher rate of diffusion as compared to the second example. Now the is second factor is size of the molecule. Size of the molecules. It is very simple and obvious, and it is very much related to common sense. The smaller the size of the particle, small size, there will be greater the rate of diffusion. The larger the size of the particle, there would be less rate of diffusion. Or the smaller the size of the particle, the higher the rate of diffusion, and it is indirectly proportional. Next one is temperature. Number three, temperature. The higher the temperature would be higher, there will be higher diffusion. The lower the temperature, there will be low diffusion. But why? Why is this happening? It is happening because the higher the temperature, greater would be the movement of the molecules. And lower the temperature, the movement of the molecules will be limited or restricted. That's why. And number four is medium. In which medium you want to diffuse? As we already know about our previous examples, if the medium would become gas, there would be greater diffusion rather than liquid. Sorry, yes, rather than liquid, and more or less in solid. So most the rate of diffusion is most high the rate of diffusion is in the gas, and then liquid, and then solid. And the number fifth one is surface area. The higher the surface area is denoted by SA, there will be higher in diffusion or rate of diffusion. The low surface area and vice versa decrease diffusion, the rate of diffusion. Now the number six, solubility. how much the ions or the ingredient or molecules have to be diffused, they are how much soluble in that medium. So it is directly proportional. It is directly proportional. And the least, last but not the least, it is the thickness of the membrane. More increased thickness, decreased diffusion, and vice versa. Now, the simple diffusion, the rate or the effectivity or the stance of the diffusion or a different molecules or a substancing depending upon their different build-up or different nature is like that lipophilic is on the top one, then hydrophobic, non-polar or uncharged substances such as oxygen, carbon dioxide, nitrous, fatty acids, alcohol, steroid hormones. These are the hormones that then their diffusion took place through the lipid matrix that are lipophilic, hydrophobic, non-polar or uncharged substances. And recording water, water is small molecules with high kinetic energy, and then urea, then water, and then urea. Now the ionic diffusion. Ionic diffusion through the channel proteins. So ions, number one, they diffuse through the channel. They diffuse through the channel. Number two, channels may be open or it may be guarded by gates. This is much better. Number three, open or leaky channels. Leak channels such as potassium channel, sorry for N, gated channel open with a stimulus. Number four, gated channel that open with stimulus. Voltage gated, ligand gated, and mechanosensitive gated. It may be voltage gated. Number B, ligand, and C is mechanosensitive. Now I would give you a very simple example of a diffusion that is happening every moment in your life, every second in your body. Is the simple diffusion that you inhale or breath in a normal air. The oxygen is diffused into the blood vessels where there is less oxygen. A carbon dioxide diffuse out of the blood vessels where there is less carbon dioxide. If we make in a diagram, if this is an alveoli, there is a blood vessel around it. Every alveoli contain blood vessel, and we donate this color to oxygen, which is very much less, but in the lungs, in the air, it is very much high. And we donate blue color to the carbon dioxide, which is very much here but less in it. So the carbon dioxide, this is the blue color, the carbon dioxide, and this color is the oxygen, which is very much rich in the lungs. It will diffuse through lungs into the blood vessel from higher concentration towards lower concentration, and the carbon dioxide with the blue color, it will diffuse out from the blood vessels into the lungs against by the following the rule of moving higher concentration towards the lower concentration. So oxygen is diffused into the blood vessel and carbon dioxide is diffused out from the blood vessels into the lungs, and we exhale out the carbon dioxide, and the oxygen that we already inhaled that was absorbed in the air was diffused into the blood vessels, and our blood get cleaner or get more oxygenated. In a medical way we can say that our blood become more oxygenated after passing through the lungs. This was a simple diffusion. Now we came across through the facilitated diffusion. Facilitated diffusion, as its name shows, it should be facilitate or compensate or helped by some other thing. So facilitated diffusion is the movement of specific molecules or ions across the membrane assisted by a carrier protein. It is by a carrier protein. Direction of the movements is down, concentration gradient in the molecules concerned. As the direction of the movement is down, the concentration gradient of the molecules is concerned as from where it is needed or from higher concentration towards lower concentration, and there is no energy required as in simple diffusion. Sorry for interruption and surprise that I have already drawn this diagram due to conserve time. So this is a protein channel. This is a protein channel in which there is a molecules is moving across the cell membranes. There is an extracellular and here is intracellular. This is an extracellular and inside below it is the intracellular. Then one is a carrier proteins. These are the carrier proteins that is shown in different stages. This one and this one. This is a carrier protein. The molecules is sent into this. It will eventually come here, and it will closes from upper side from an extracellular side and opens from inside, and this molecule will eventually come inside the cell membrane. So this is a type of a sort, or this is a facilitated diffusion that actually facilitate the movement of molecules from in extracellular environment into the intracellular environment. I have shown two types of proteins. These are the channel proteins. Uh, from which color I should donate? Uh, take this color here. This one is channel protein, and these are the carrier proteins. So what is the basic or major difference between these two types of proteins? This channel and what about this carrier? So carrier proteins carrier proteins actually bound to a larger molecule. They bound to large molecules and change their shape so molecules can diffuse through. There is a shape change, and molecules move into intracellular. While as the channel proteins provide a water-filled pores for charged ions to pass through. It is just a water-filled pores having allowing water-filled pores allowing the charged ions to pass through. Charged ions into intracellular environment. So for we have read about the simple diffusion and the facilitated diffusion. So what is the main basic difference between simple diffusion and the facilitated diffusion? Simple diffusion versus facilitated F FACILILATED facilitated diffusion. Sometimes it's not easy to remember all the spellings at once when you are fighting with your mobiles and remembering what you have to talk to your students. It's very difficult. So simple diffusion, it is a concentration gradient. Oh, should change the color. It is concentration gradient dependent. It is also concentration gradient but with through carrier proteins. You should at least know about the basics or basic difference between simple diffusion and facilitated diffusion. So you can write or attempt one question with 1 mark or 2 marks in your annual and in the in your practicals. Number two, this is number one, this is number two. Rate of diffusion. Oh, we already discussed about the factors and all other things that were diffusion, but in this comparison, uh, it depending upon the concentration gradient again, steepness of the concentration gradient, that is a proper or steepness of concentration gradient. I already explained about the steepness of concentration gradient. When there is a much more higher concentration in the one region and another region there is a low concentration, the rate of diffusion would be higher, as we have already discussed. And in this facilitated diffusion, it purely depends upon the saturation of carrier proteins or the concentration of Carrier proteins. If there would be more carrier proteins available, there would be more facilitated diffusion. Carrier proteins. I hope so okay, that you have learned this transport across cell membrane concept and gone through the facilitated and simple diffusion. I tried to make it very much easier and simple for you, but if still there are some questions or misconfusions or anything, you are more than welcome to ask. But I request you to listen at the end of the lecture, the whole lecture, and at the end, give me the your questions or whatever you want to do. But questions should be in a group so that I can comply on the reply with efficiently. Thank you very much.